We are live.
Good evening, all. Welcome to iFocus Online, episode 313, the fourth in the Back to Basics module. Today, we have with us Dr. Subhabrata Chakrabarti from LV Eye Institute, Hyderabad, to speak to us on essentials of ophthalmic genetics. Sir is a molecular geneticist, largely trained in India and at the National Eye Institute, fellow of the Association for Research in Vision and Ophthalmology. Currently, he is the Associate Director of Research at LV Eye Institute, Hyderabad. His major work is on functional genomics of primary congenital glaucoma. His work initiated the bilateral grants with Brazil, Tunisia, Portugal, Australia, and the United States of America to understand the nature-nurture dialectics in the primary congenital glaucoma. A research recognition award of World Glaucoma Association was awarded to him, and he has young affiliateships of the 3 Science Academies of India and the World Academy of Sciences. He is also the elected member of the Guha Research Conference, Associate Editor of Investigative Ophthalmology and Visual Sciences. He's an expert on the ClinGen panel, accurately curating the widely used ClinVar database. We welcome you, sir, and we'll be highly enlightened today after your talk. Over to you, sir.
Thank you. And at the outset, let me thank Dr. Santosh Honavar and the team of CFS for this kind invitation. I remember I had spoken at iFocus last year, and it's again coming back again as a very good opportunity. So what I'll be doing over the next 40 minutes or so is to take you through the essentials of ophthalmic genetics, which is the topic that has been assigned to me, and which is one of the priority areas, not only for geneticists, but also for clinicians and also from other fields of science, because genetics again forms the basis of human life. So why is it important? If we look into the World Report on Vision, everyone, as they live long, would have some eye conditions. And we know that cataract and refractive surgery are the 2 major blinding conditions, costing about $14.3 billion US. And at least 2.2 billion people live with a vision impairment. 1 billion of these cases account to any of these conditions, and some are even unaddressed. Age-related eye conditions, as the demographics are changing, senescence is on the rise. Age-related eye diseases are also on the rise, and we see AMD and glaucoma and diabetic retinopathy to be on the forefront of these age-related eye diseases. The interesting fact is that there are risk factors which are modifiable, like the lifestyle-related factors or environmental factors, but there are conditions which are non-modifiable, like aging. We cannot reverse aging, the genetics, the basic genetic format of any human being cannot be reversed, ethnicity cannot be reversed, and none of this constitute the risk factors towards these diseases. So to give an overview, what we are talking about, so the caveat is that we are talking about a nucleated cell, which has the chromatin, the chromosomes condensed into a chromatin network, and these chromosomes harbors DNA. DNAs are the deoxyribonucleic acid. There are 4 such amino acids: adenine, guanine, cytosine, and thymine. A always pairs with T, C always pairs with G, and they are again bonded by the sugar and the phosphate bonds. A portion of these signatures account for amino acids, and these amino acids form proteins, and this constitutes only 2% of the entire genome. So when we say the genome is a stretch of this ACTG across all the chromosomes, right from the telomere to the telomeres of all the 22 autosomes and the 2 sex chromosomes. And that length is now estimated to be around 3.06 billion bases. When I say base, each of these is a base. And within that, you can imagine that it's only around 2% which constitute genes, which carry out functions. Genes code for they transcribe to RNAs, and then further translates into proteins. Proteins carry out the cellular functions, and these functions are what we see being enacted in terms of phenotypes. If we look into genetic disorders, now we are moving from what the fundamental basis of genes are. And if there is any defect in those genes, that leads to disorders which are genetic. And genetic disorders are usually inherited. If it is at a prenatal or antenatal stage, they're usually very lethal and they are eliminated. So we don't have these proportions. But there are diseases which are congenital in origin to late onset, early onset and late onset. And this could be anywhere between severely disabling conditions to mildly disabling conditions. And this is the spectrum which we call a clinical disease. Within that, there are other variations in the human genome that are neutral and do not contribute to any specific phenotypes. So this entire stretch of A, T, C, and G repeated at tandem constitutes the whole human genome. And this genome further carries out the functions based on the nature of how it has been inherited, how it has been subjected to other environmental conditions, and that is how it reflects in the phenotype. But there is a problem, and the problem is that molecular mechanisms, because of or due to these changes, are not very simple. They are very complicated, and the complication arises because there are clinical heterogeneities. That means within a conditions, you can have multiple types or multiple phenotypes of the same condition. But more added to that, we have genetic heterogeneity. That means that a same gene may not cause the same disease all the time. It could be locus heterogeneity, meaning thereby that there might be multiple genes which will be causing a single disease. Or there is allelic heterogeneity, which is that within a gene, there could be defects at multiple regions of the gene, resulting in a particular disease. It could be due to reduced penetrance. So sometimes one could harbor those changes but not manifest the phenotype, or there could be differential expressivity of the genes, meaning thereby that the number of copies of the genes might be different depending on the condition or the phenotype. So as I said at the outset that genetic diseases are usually inherited. And when we talk about inheritance, inheritance can be of any of these kind. If it does not skip generations, it has two different alleles which equally contribute to the disease. It's an autosomal recessive disease. Parents are usually carriers. They carry one copy of either of the allele, but the child carries both the alleles. So these are autosomal recessive diseases. And usually these are consanguineous, meaning thereby these kind of a structure comes out of consanguineous marriages where there is a marriage between close relatives. It could be first degree or second degree cousins. It could be uncle, niece. It could be any of those. So these are autosomal recessive diseases. And then there could be X-linked diseases, which are usually on the X chromosome. The females are usually carriers, but the male, which has only one X chromosome, manifests the disease. It could be autosomal dominant. That means it could be in any of the either of the 22 chromosomes, 22 autosomes. And autosomal dominant disease usually does not skip generations, as you see in the other previous conditions. It doesn't skip generations because you need only one allele. You don't need both the alleles to manifest the phenotype. Then there are cases of de novo mutations. That means mutations that are not manifested in the parents, but still it occurs in the child. And then there could be tissues which could harbor various changes, and that would again result in an affected tissue. So in starting this complicacy, we have seen a major paradigm shift. One is because of the defined goals and the available technology. So earlier, the approach was more of a forward genetics, where one actually studied the function or the phenotype of the function of the phenotype, particularly in terms of disease. And then they try to identify the gene within, they're responsible for the function, clone that gene and localize it to the chromosome. And a classical example of that would be, let us say, if I'm identifying genes for retinitis pigmentosa using a forward genetics approach. So I would like to understand what the phenotype is, and if it is a problem with the rods or cone cells. So I would try to find what are the genes that are present in rods and cones, then try to clone those genes, identify the mutations, and then see where those are localized in the chromosome. As opposed to that, currently the approach is reverse genetics, where we take a model-free approach. That means we do not restrict our understanding to that particular tissue or that particular phenotype, because there is no one single gene that can cause a disease. Diseases are caused by multiple genes with varying magnitudes of effect. So one first localizes those specific genes on the chromosomes. So what is done is usually a linkage analysis. If it is a family, I'll come to those in details, or it could be through whole scanning of the whole entire genome. So one finds where or on which specific chromosome the affected genes are. Affected genes meaning thereby that genes which has an affected variant or a different variant which is linked to the affected phenotype. And then they clone that particular variant, identify the gene, and then try to assess its function. So the function, so here, if we take the same retinitis pigmentosa family, so we would scan their entire genome or entire chromosomes, try to find out if the genes are particularly in one chromosome or multiple chromosomes, then assess their functions and see which one of them are more appropriate. So this is the phenomenon of reverse genetics. So in conventional approach, one would actually like to do a kind of a family-based linkage study as, sorry, it's moving the other way. One would try to assess the linkage. That means whether that particular, as I said, in a reverse approach manner, that if we scan the entire chromosomes, are there specific chromosomal region that harbors the mutant allele and that segregates only with the affected but not in the unaffected? And here we are trying to characterize mutations, meaning thereby we are trying to characterize variant which have a very large effect, because we do not want to have that in the controls. So the effect of that, or the allelic effect, is very large. On the other hand, if we have diseases which are age-related, like AMD, or like age-related glaucomas, or diabetic retinopathy, where we may not have a proper family history in this manner, where the samples would be available from all the 2 or 3 generations, the approach is to do an association study, whereby we choose individuals who are affected and match them to controls from the same ethnic background, matched again for age, gender, and ethnicity, and scan the same genome and try to find out that are there specific signatures that are present in more frequency, where the prevalence of those frequency of those variant alleles are more in cases which we do not see in controls. Controls will have those, but they are relatively lesser. And so within those regions, then one tries to map genes. So the approaches in this two is that in one, we are trying to find the effect of the allele, which is very large. Here we are finding alleles of smaller effect. On the other hand, in terms of frequency, these will be rare changes or rare variants. So the frequency will be low in family studies, whereas the frequency of common variants will be very high in the populations. So these are the two major approaches that we use in terms of identifying genes for specific diseases, be it inherited diseases where we have proper family history, or complex diseases where we do not have a proper family history. So from a clinical perspective, what we are looking into is that an undiagnosed patient reports to the clinic. So there is a structured phenotype analysis, and sometimes it could result also in deep phenotyping. And then one could go in for the genome sequencing and the sequencing could be of all genes or partial genes, and along with the family members, then the data is analyzed, and there is a diagnosis given based on, based on what the findings are. And though based on that findings, there could be either a change in management in terms of the therapy, in terms of monitoring, or in terms of planning, even in terms of genetic planning as in genetic counseling. So this is more of a conventional approach. So I was talking about linkage analysis. So if we have families, we scan markers across the chromosomes, try to find out those specific markers which segregates only in affecteds or partially in the affecteds, but not in the controls. So controls are devoid of the shaded markers. And then we try to find out which are the ones where between them, we try to find out regions of commonality. And within those regions, we try to see if there are genes that harbor mutations. So these are technically called haplotypes. Haplotypes are nothing but series of alleles on a haploid chromosome, on a single chromosome. And we try to find out what markers are. And then based on that common region, which is shared across all affected individuals, we try to narrow it down and try to find out if there are genes. Then that region is sequenced. We try to find out if there are changes, which are again present specifically in the cases, not in the controls. And that segregates only with the affected phenotype, not in the controls, as you can see in the controls. So in the cases, you see the father is affected and the two children are also affected. They have a specific band pattern, which is not there in the mother who is unaffected. And then we also check it in the normal controls with the father and the others as father, mother, and the child as positive control, and the others as negative control. You see the pattern. The mother who is negative doesn't have the second band, and all the controls do not have the second band. So this is a very straightforward approach. We also try to see whether that region harboring the protein, the corresponding amino acid, is conserved across different species. If it is conserved, then obviously those regions becomes region of importance. Using bioinformatic tools, now one can model those proteins and see if there is a structural or a functional change between the native protein, that is the wild type protein, and the mutated protein at that point. And so with this strategy, one can map genes for multiple diseases. I'll give the example of primary congenital glaucoma because we have been working more on that. As you can see in primary congenital glaucoma, again, these many chromosomal regions were mapped by linkage, but only two genes, that is the cytochrome P450 CYP1B1 and LTBP2, or genes were identified to harbor mutations. CYP1B1 happened to harbor majority of the mutation, and a very minor proportion of mutations were there in LTBP2. Whole exome sequencing, which is a relatively newer technique, identified another gene, again with lower frequencies. And then there were screening of candidate genes. That means these are genes in other glaucomas, not necessarily in congenital glaucoma, and they were also found to harbor mutations of varying frequencies. And if you see the distributions, then how do we finally conclude that these are the most responsible genes? Because we are talking from a point of view of clinical ophthalmology. So we try to see what's the proportions of these mutations in other populations. And also we try to assess the functions of those individual mutations. Does it, like for instance, CYP1B1 in this case, we try to see whether they are responsible for, and because CYP1B1 is an enzyme. So how does the catalytic activity or protein stability changes, given that there is a change in the DNA sequence of CYP1B1? And does it actually localize, or is the localization affected within the cellular milieu? So if these functions are affected, that also conclusively proves that there is some element of pathogenesis that is linked to these variants or mutations, and that in turn might be responsible for the disease. The advanced approach, and these were all approaches which were conventional, where single genes were involved. But later it was found that it is not one gene that is causing a disease. Each disease is responsible or is attributable to multiple genes with varying magnitudes of effect. So in that 2x2 table, what I showed you that in familial diseases, we had a very large allele, or a large allele effect, and a common allele had a larger frequency and a small effect. But as we go along, you would see that it's not one gene or one allele, it's varying alleles of varying frequencies with varying magnitudes of effect that is responsible for the phenotype. So coming to complex disease, we have multiple phenotypes. There could be effect of environment, and collectively that produces the disease state. Now, each of these phenotype categories is contributed by different genotypes or different genes. For instance, if we take glaucoma, for instance, that adult onset glaucoma, we have intraocular pressure, we have retinal nerve fiber layer thinning, we have cup to disc ratio that's changed, and there could be multiple phenotypes. Let us say we are talking about AMD. Again, we'll have different phenotypes that collectively contribute to AMD, and then there could be environmental variants or environmental determinants like smoking, alcohol, etc. And each of these collectively in the background of this in the genetic milieu would contribute to the disease state. So what happens is the advanced approach then led to identification of not genes only within the coding region, but across the genome. So various types of markers were selected. So the most classical was the 1 million marker of small repeats of DNA across the genome. And people then tried to narrow it down to a single marker or small sets of markers. It was like fishing a needle in a haystack. And then they came to a conclusion. And later on, what happened was sequencing. There was a sequencing explosion where the sequencing techniques advanced. And as people could sequence more and more, the costs also parallelly came down. So from gel-based system to capillary system to massively parallel sequencing, which is our next generation sequencing, to single molecule sequencing. So as you can see, proportionately, this has gone up and the corresponding costs have come down. Now, with this data, what do we do? So what we have is a different kind of a strategy, because now we have different mechanisms of generating a very comprehensive data in terms of the biological data. So the approach that we take or others take is that there are candidate genes, and candidate genes are those which are mapped by linkage analysis, which harbor mutations, which are known and which are characterized to affect functions. So one initially characterizes candidate genes. Then if there are cases which do not harbor mutations in candidate genes, one then goes in to understand them either through genome-wide association studies or through whole exome sequencings. And between them, they try to see what are the interactions that are happening. And based on the functions, they try to assess the genotype-phenotype correlations in order to understand disease pathogenesis. I'll explain it in more detail as we go along. So before that, because now next generation sequencing is a big buzzword, so I would also like to caution everyone that the quality control measures need to be very appropriate and very stringent, because as we see an avalanche of data, it also throws up things that may not be necessary, that may not be important, that may not be relevant. So for instance, when we do whole exome sequencing, and when we say whole exome, that means we are talking about the 2% of all genes in the genome. And that constitutes, so the genome has close to 16 to 20,000 genes. So we are talking about sequencing all those 20,000 genes at one go. What one needs to understand when we are sequencing these things is, are all the genes being sequenced properly? Because we are looking into multiple genes of various sizes, various structures, and they are not linearly arranged. They are arranged in the chromosomes in various forms. So when they are kind of disentangled and then they are sequenced. So they should give the entire stretch in one go. Like for instance, one needs to see how much of those genes are covered when we do whole exome sequencing or even whole genome sequencing. Like, is it 100% or is it less than 100%? Like here, you see that across some genes, this is only a snapshot just to make you understand, that for some genes, you may find 100% sequence, 100% coverage, but for some, like it is 92, 96%, and so on and so forth. The reason for this is that these genes are sequenced by certain stretches of DNA, which are primers. And these primers also bind to other regions, because when we say that it is a stretch of DNA of ACTG of 3.06 billion base pairs, so obviously there are regions which are common. And when a primer of these gene also finds a complementary sequence here in the region of the other gene, it also falsely binds here. And so one is unable to distinguish sequence. Or sometimes the primers of these genes may not allow the primers of these genes to kind of amplify together in molecular reactions, which we call PCR. And so part of this region remain unsequenced. So it is very important to assess how much of these regions are sequenced, because those regions then need to be separately sequenced. Number one. Number two, how much of the region is missed? So like, for instance, here in one of the genes, we find that most of the regions are covered, but there is one small region of 148 base pairs, which is missed. And then one needs to assess one needs to kind of employ a different strategy to sequence that. The next thing is the sequence done uniformly? So these are different samples, and these are the reads of those sequences. How many are on target? What is the depth of the sequence? And how many are uniform, in the sense that how many are a very reliable sequence? Like here, when it goes down to 50% or so, these are unreliable. And these needs to be, again, either resequenced, or if the sample was not proper, or the DNA starting material was not of good quality. And how do all the samples provide the same number of variants? That is another thing that needs to be taken care of. So, for instance, one of these samples had a low number of variants. Now, it could be possible that there are some samples with low number of variants, but that needs to be assessed by doing a resequencing. So now, having understood very broadly, I mean, there are a lot of other nitty-gritties which I'm not going to. Having understood very broadly about this, let us go into the whole exome sequencing and targeted sequencing, where we are talking about the rare and common variants together. So when we talk about whole exome sequencings, and this is a very good method to identify all genes in one go, one needs to see whether the depth of sequencing is appropriate. By depth, I mean that all genes are evenly sequenced across the genome, and they have equal power to represent the sequence accurately. So if there are samples which are dipping, so that means those needs to be repeated. And why it has to be done? Because when we sequence an exome, what we find is that more than half of the sequence is synonymous, meaning thereby there are changes, but that doesn't change an amino acid. So 3 bases, that is one of the 3 letters, constitute an amino acid. And if that letter changes in the first or second sequence, that changes an amino acid. If it changes in the third place, it doesn't change an amino acid, the amino acid remains the same. Now, the question is where the change happens. It then might happen in the RNA. So that's a very long drawn shot. I'm not going into that. Just for your broad understanding, that when we do a whole exome sequencing, more than half is not required immediately. Another major chunk is missense. That means there are changes, it changes the amino acid, But all of it need not be functionally changing the either the structure or the functioning of a protein. So again, this is another chunk which needs to be identified properly. And then there are copy number changes, insertion deletions, and nonsense mutation. That means mutations which actually terminate the production of a protein. So the protein is truncated and it cannot function. So these are more reliable changes, or what we call as mutations due to loss of functions. And in a real life scenario, what you would see is, for instance, in primary congenital glaucoma, like when you do a sequencing of whole exomes, you find anywhere between 50 to 75,000 genome, 75,000 variants. And now, since congenital glaucoma is recessive, we are looking into homozygous changes, as I explained in the initial slide, changes which are homozygous in the patient and heterozygous in the parents. So now you see from 53,000 or 75,000, your number comes down drastically. Now, many of these variants will also be present in the general population, so that would not really have a functional implication, so those are removed. Out of them, how many are coding? Because as I said, most of them will be either they will be synonymous, or they can also be in other regions which are of the introns, which may not be immediately responsible for certain functions. So then, as you can see, the number actually drastically decreases. So then there are non-synonymous, then there are pathogenic variants and the frame shapes. So this is how we kind of assess what could be the potential changes when one does a whole exome sequencing. So using this strategy, as you can see, we find that in congenital glaucoma, don't go on reading the percentages and genes, but what the take-home message is, if we do only candidate gene screening, almost 58% of the purple region remains unanswered. That means 58% of the patients do not harbor any changes in the candidate genes. When we take other genes, targeted sequencing, like we take the whole genes in the anterior segment and other regions, what we find that from 58%, it drops down to another 10%. So the unknowns are around 48%. When we do whole exomes, it doesn't completely eliminate, but it still brings down from 58, it comes down to 44. Now, why this is so, I'll come to later on. And then we need to kind of assess whether those are actually expressed in the appropriate tissues. Now we are talking about congenital glaucoma. We would like to see whether they are expressed in developmental tissues, because these are genes which need to be expressed during development for proper function, proper ocular development. And so those are the genes that will be responsible, or that would be primarily responsible if they are expressed at that point of time. For instance, genes which are not expressed during development are unlikely to have any functional implications in the causation of congenital glaucoma. Now, you saw that after doing all of this, there was around 40% unknowns. And that is true not only in glaucoma, even in another study done on retinitis pigmentosa, using the same strategy, they found that again, a large portion, around 37%, is unknown genes. And there are now diseases now which we understand and which we are doing as to how to reduce this gap, and I'll come to it later on. And those gaps are because there are cellular and molecular mechanisms that are taking place due to the interactions of genes, not by individual genes. So when we look into those, genes, as I said, they have overlapping functions. And so one gene, as you see, cannot be responsible for one function. Like, again, this is from a glaucoma perspective, like intraocular pressure has been assigned to so many genes, central corneal thickness to even a large number of genes. And then there are genes which are shared between the two. Likewise, optic cup area assigned to so many genes, cup to disc ratio assigned to these many genes, disc area to other genes, and then there are genes which are shared across them. Now, the problem is, once these are shared, these are not necessarily mutations. These are common variations that you also find in normal individuals. Maybe the proportions of these variants are relatively higher in the patients. So when we do that in congenital glaucoma, we find that there are multiple such variants in specific genes which contribute to risk, which are in red, and then those which are related to protection, both in anterior segment disease-related genes, glaucoma-related genes. And so these collectively, along with the mutations, would result in the phenotype. And one then kind of classifies them based on their functions and comes to this kind of a arrangement. So what we find is we find rare alleles, which cause of the rare mutations, which are responsible or which segregates in the family. Then we have variants of intermediate frequency and common variants. So if you look into genes in congenital glaucoma, like CYP1B1, LTBP2 TEC, MYOFOXY1, they are harboring rare changes. That means these are mutations. But then there are other changes in other genes which have an intermediate frequency or very low frequency in the which are common variants, which have a high frequency, low effect, but high frequency in the general population. So the collective interplay of all of these variants and mutations are responsible for the phenotype. So if one tries to kind of assess their functions based on a network, what you see is that many of these, they are related by functions, but those, there are certain genes which are not related to this main network of genes, so they may not be directly implicating the functions. So like this also, one can eliminate genes which may not be implicated in the disease. So this is how the distribution looks like. So you have rare variants, you have significant proportions of common and intermediate frequency variants. So when you collectively do that and then try to assess how many of these interact with each other, we find that from that proportion of 44% or larger than that, we have a significant number which are co-occurring, which means that these alleles, they interact with each other. So these by themselves, so single alleles within those genes, candidate genes may cause disease. Single alleles within anterior segment dyskinesis and other related genes may cause disease. But interactions of these genes, that also forms a significant proportion, thereby reducing the number of unknowns. And those are, again, the blue ones of varying frequencies compared to controls of various regions. Now, when we are looking into intermediate variants and common variants, we also need to assess controls. And this is one of the most important things, particularly from a clinical perspective, because we need to have controls who do not harbor those for the variants in the cases to be implicated in the disease. So what I'm talking about is that within our cohort of controls, we would not like them to harbor those changes. So they'll be of zero frequency or very minor frequency and should not be harboring a very high frequency in other populations as well. One of the difficulties, particularly, and this is for those clinicians who actually use service providers' data and who sends their samples to service providers, sometimes they compare that data because they do not have appropriate controls. So they compare to a global database called the 1000 Genome or the GenomeAD, which has some relevant populations like populations from Lahore, from Gujaratis who are settled in Houston, Tamils of Sri Lanka, Telugus from Andhra, Bengalis from Bangladesh, and et cetera, et cetera. So when you collectively see these data, and now let us, we actually tried to see whether these are actually true data. When we saw our population cohort data taken from Andhra and Telangana, what you see is that the diversity of this data is very large. The proportions of the alleles are low compared to all of this population. So you have less number of alleles, and most of these are of the same magnitude, which meaning thereby that most of these are related individuals and do not implicate that the observed variant to be pathogenic. What do I mean by that? So if these data are compared, if you see just the number of bars here, and so if your sample or your patient sample has a variant which is away from these bars, it is only compared only with the 6 or 7 bars, as opposed to if you take populations from your region, ethnically matched, gender ethnicity matched controls, you would find a wide variability among that. And so you would see that whatever you have found based on the genome ad or 1,000 genome may not be true. So it's always important to kind of compare your data to the controls, whatever are present, or whatever ethnicity the patients belong to. And to give further example, like again, from a congenital glaucoma perspective, so these are the different patients. I'm just sharing a snapshot, and these are the different genes. So the red means they have homozygous alleles. The blue means they are heterozygous, and the yellow means they are compound heterozygous, which means that if it is red, that means they have 2 affected alleles from this particular gene. Let us say this is CYP1B1. If it is blue, that means they have 1 particular allele in CYP1B1. If it is yellow, that means they have 2 different alleles, not the same alleles, 2 different alleles, maybe one in the beginning of the gene, one other towards the end of the gene. So those are compound heterozygotes. Now, if you see the patients along the columns, you see that most of them harbor multiple alleles to cause the phenotype. So the point that I was trying to drive in earlier, that the diseases are not caused by single genes, but there are multiple genes, multiple alleles with varying magnitudes of effect across different genes. And so these are different genes, and each of them have a different profile. So collectively, if you see, they have a different profile. Now let us see with respect to the controls. Even for the controls, there are controls who have, although the numbers are much lesser here, but still there will be controls who would harbor a similar profile across different genes. So when we find a match to the profile, then that profile needs to be eliminated, because that's also there in the controls, and that might not be responsible directly for the disease pathogenesis. When we take other genomic databases, like the 1,000 genome or the genome ad databases, what you see is that majority of the subjects in those databases will harbor single variations in one allele. The number of alleles or pathogenic alleles will decrease over a period of the number of genes. For instance, patients might have multiple alleles across multiple genes, but controls will not have multiple alleles across multiple genes. And so likewise, also, one needs to eliminate what are the different unique signatures that are present in the patients, which are not there in the controls. In order to do so, now there is a mega project that is Pangenome Reference, which is planning to map the entire global population of normal controls, and then they are trying to delineate signatures which are exclusive to the controls, so that given that those signatures are present, they may not harbor any disease phenotype. Even the role of ancestry, when we talk about mutations, the role of ancestry is very important. As we know, populations originated in Africa, and then there was a migration out of Africa. So when we look into populations which have migrated out of Africa, and we find variations or mutations in a particular disease gene, there will be certain commonalities, and there will be certain signatures which will be common, because as populations move out, well, there will be recombinations between them, but that recombinations will take a long time to disappear. So to cut a long story short, like in primary congenital glaucoma, if we look into CYP1B1 mutations, North African populations, South Arabian populations, Indian populations have a kind of similar mutation profile, and similarly Indians and South Arabians and Brazilians. Now, populations moved out of Africa, moved through the Middle East to India, to Southeast Asia, and there was another clade that moved up from Europe and went all of the way to America, North America, to Latin America. So the number of recombinations that happened over there are much more as populations diverged from the mainland to the other lands. So which means that the frequencies of mutations gradually comes down as they go to from Africa to Saudi Arabia to India to Southeast Asia to all the way to Australia. So if you see in Australia, the frequency of CYP1B1 mutation is much low, the number of mutations are low, and the signatures are different. Likewise, it happens in Brazil and other parts of Latin America. So from a clinical perspective, what it means that no matter which population you come from, you have a same signature. That means if we talk about this mutation, no matter whether you come from Ecuador, Morocco, Saudi Arabia, or India, you are on the same signature or the haplotype, and likewise for other populations. So from a diagnostic point of view, if a patient harbors any of these 4 haplotypes, you would be certain that they also harbor any of these disease-causing mutations. So that is again another way of kind of shortlisting patients for mutations. And likewise, we have seen that based on ethnicity, based on language, patients almost share signatures and mutations. The same thing with respect to LTBP2, that there are much lesser mutations in the Indian context because LTBP2 is largely restricted to patients in Pakistan, Iran, and the Gypsies. So I'll skip this part, but we'll go to pathogenicity. So now once having established that these could be potential variants, how do we establish pathogenicity? So to establish pathogenicity, there are certain guidelines, and these guidelines are provided by an NIH initiative called ClinGen. And what ClinGen does is to kind of ClinGen is a group of individuals across different subspecialties, and they try to curate this data and then put that into the ClinVar database, so that as a clinician, if you're trying to see whether the mutation is pathogenic or not, you don't need to do all those exercises, but you can just look into the database because these are curated by experts based on certain parameters. So I represent, as was introduced, I represent India in ClinGen, and we have different guidelines for providing a variant or for ascribing a variant as pathogenic. And these guidelines are all there, all available. And there are certain scores which we give, provided depending on the nature of the variant, depending what type of transcripts are there and what are the corresponding changes. And we also look into the functions and whether the functions are really important for that variant to confer a disease-causing phenotype. So using that same model, what has happened now is that there were a lot of random variants which were connoted as mutations. And that has created a lot of problem because many of these are not disease-causing. So when we started our work and we are part of the glaucoma panel, what we found is that myocilin and CYP1B1, the 2 major genes, have only 20 variants, which are, or 20% of the variants, which are disease-causing. So if you go into the database, there are 279 variants, but we list only 47, out of which only half of them have sufficient evidence to assess pathogenicity. Likewise, in CYP1B1, there are only 40 variants which are pathogenic. And this is the summary statistics that you get in ClinGen. And this is more from a clinical perspective, that if you would like to know what are the actionable alleles, you will really get in from this database, which are the ones which are responsible for pharmacogenomic interventions, that also you'll get, which are the ones which are pathogenic, and so on and so forth. Now, if you remember the first cartoon, there was something missing here from the undiagnosed patient who came in, had his phenotyping done, did sequencing, the data was shared, and diagnosis was given. Now, because of our understanding of the genome, there are additional data using multi-omics approach that actually complement whatever data is available from the genomic sequencing and from the clinical perspective. So collectively, that data now enriches the diagnosis, and that helps in guiding therapy and management. So for instance, I'll give one simple example. So I'll quickly go through considering the time. Like CYP1B1, we have interactions of promoter and coding variant. So there is a variant in the promoter region that interacts with this. It reduces activity of CYP1B1 by 90%. It disturbs the binding of other developmental genes. And from a clinical perspective, if you follow these patients post-surgery, they have a poor outcome in terms of IOP control as opposed to those who do not harbor these variants. Similar way, there are cases of diagenic inheritance where the parent carries one affected allele, or mother carries the affected allele of another gene, and they are asymptomatic. Child carries both, becomes symptomatic. And what happens is, because of that interaction reduces the protein functions, both in terms of their amount and both in terms of their cellular localization. So likewise, these are other examples, starting from serine protease, where you knock out those genes, see what happens in the trabecular meshwork of mice compared to controls. You see that trabecular meshwork becomes hypoplastic. And even if they are rescued, the IOP is still not normal. IOP still remains high. Similarly, another example from GLIS1, again, the same, the GLIS1 gene, when you knock it off and you see the effect, the trabecular meshwork is always hypoplastic. Now, when we talk about molecular mechanisms, it is important that we demonstrate that given that there is a mutation, there is a different profile of functions. But when we restore back, that is when we artificially or give back the corrected genes, it should again resolve its functions. That is where we establish this kind of molecular mechanisms. But from a clinical perspective, one needs to do a thorough assessment of genotype-phenotype correlations. And in primary congenital glaucoma, what we have shown is that there are specific clinical parameters which are strongly correlated and are like intraocular pressure at presentation to final follow-up, cup to disc ratio, and corneal diameter, and photopic negative response. So parameters of photopic negative response are strongly correlated to certain clinical parameters in cases that you do not see in individuals who do not harbor these changes, or PCG patients who do not harbor these mutations. So these kind of assessment also helps the clinicians to kind of understand the nature of progression of the disease, the type of therapy they would respond to, and so forth. And then with the appropriate tissue, one can understand the protein profile, understand how they interact in the cellular milieu, and how these proteins lead to the phenotype. So some of these proteins are leading to developmental defects, some to cellular deaths, and collectively how they affect the primary phenotype of the anterior chamber defect and trabecular meshwork defects resulting in the disease. So these kind of approaches, now why I'm coming towards the end. So these are multi-omic approaches where one can actually see the interactions of the genes and proteins and try to assess what these functions might be. And so currently there is a newer thrust to understand mutations in the background of common diseases, and one then tries to understand how this single gene variants and common variants, how they interact and result in a phenotype. And as one can see that across genes, the effect size of the mutations are much larger compared to the effect size of the common variants, but collectively they produce a larger effect. So one needs to discover genes, understand interactions, understand the physical interactions, that is how the proteins functions, understand the functions through models, and collectively assess how omics approaches can be integrated in the background of clinical, surgical, and rehabilitation options to provide a better disease management and towards a better understanding of pathogenesis. In terms of complex diseases, one like glaucoma, AMD, DR, etc., one can use these multiple approaches, both from perspective of imaging, perspective of genomics, and all the risk factors taken together, and use that to generate some kind of a risk stratified treatment for a better health outcome. So going forward, there are now approaches of clinical genomics to reverse phenotyping. So one can actually see just based on the reverse phenotyping is basically given that the patient has a genotype, what would be the possible phenotype? Then one needs to differentiate between genetic screening and testing. So genetic testing, for instance, one would like to go on for predictive testing only when there is an actionable allele, or diagnostics testing if that actually changes the management pattern. So based on the type of disease, one can then choose what approach to take for a better clinical outcome. At LVP, we use now multiple approaches of not only studying the nuclear genome, but also the mitochondrial genome and the crosstalk to understand how the disease phenotype is implicated. And there are multiple avenues from which these data are available, like data from the NIH registries. And lastly, I would come to two things which I'll not talk today, is that of epigenetics, systems biology, and the Mendelian randomization. So since there is an avalanche of data, which I talked earlier, so we would like to kind of assess all of this in the background of random segregation of alleles based on the disease outcome, and collectively assess them through some systems biology approaches. And this systems biology approaches then teases out what are the best parameters to explain a disease progression or a disease causation. For undertaking genetic testing, there are defined guidelines. I'm not going into this. These are AO guidelines, which I've talked about last year. And lastly, we need to strike a balance of doing all these genetic tests based on the opportunities it provides and how it can help. Because these are opportunities also for discrimination, for eroding privacy, confidentiality, there is high cost, rising disparities. But at the same time, this accelerates drug development, it simplifies clinical trials, adverse events, and customizes drugs, and there is effective disease treatment strategies. So one needs to strike a balance, given that there is an avalanche of data. So lastly, I would like to thank all the patients and volunteers, our team, collaborators, and the funding agencies. And thanks for your kind listening. Thank you.
Thank you so much, sir. Wow, I can understand how and why the field of genetics is like a vast ocean and so interesting. And I'm sure many of our viewers would be interested in genetics. And if they are, I'm sure they will contact you for the same. And I'm glad also that this talk is going to be online, because I think just to absorb the things and everything, I think it needs to be seen again. So yes, sir. So thank you so much for that talk, sir. So we have a few questions. So I have a practical questions, because we deal with retinoblastomas. I think you talked about PCG a lot. But as ocular oncologists, we deal with retinoblastoma. Can you just briefly tell us about the genetics of that also, and how, like, clinically we should apply the genetics when parents ask us, especially if they are trying to conceive a second child. So genetics about that, sir.
So retinoblastoma, as you know, the RB gene is one of the major implicated genes. And you have this Knutson's hypothesis of two hits. So you have one change which occurs in germline, that is, it is inherited, and another change which is acquired. And so in retinoblastoma, what happens is, if there is both the changes are there, then one can design the treatment strategies accordingly. Problem is if these are not due to RB1 mutations, because RB1 mutations and its management is fairly straightforward. And finding RB1 mutations also has different simplified approaches now. Like earlier, the sequencing of the entire RB1 gene was a bit of a challenge. Now there are multiple approaches to understand or identify what the changes are. But if it is not a RB1 driven mutation, then one needs to find out the other genes. And there, the strategy would again be to use a model-free approach. Like one can do a whole exome first and kind of identify if there are genes other than RB1, and the characterization of the somatic mutations, because all these cancers, they do harbor somatic mutations. So the strategy would be the same, like finding the potential variant, the corresponding somatic variant, and then trying to see, given that they interact or given that they co-segregate with the disease phenotype, does it actually cause the disease? Does it have any bearing in individuals who do not harbor the disease? So, and then trying to assess pathogenicity based on the strategies that I mentioned.
Okay. Yeah. And so another question, if they are trying to have a second child,
Yes.
in utero, should the child be undergoing a coronary sampling and then a genetic testing on that sample, or just a screening should be okay, like once the child is born?
There are two approaches and there are two schools of thought in this. So one approach is about genotyping the parents very efficiently, because ultimately the parental genotype will recombine to get into the child. So once the parents are sequenced at length, and if there is an evidence that there could be a potential of the child harboring the mutate allele based on the previous child's genotype, so then obviously one can control it in utero. But the other thing is, if you do not find anything, so then that is where, again, it's a question of resources also, because whether one can do a whole exomes on the entire family and then provide a kind of a diagnosis. So, yeah. But the crux will be to identify the changes in the parents and assessing whether those will go into the child based on the entire genetic profile of the first child.
So I think we have a few more, but I think you have covered some of them in your talk regarding the early, like the basics of genetics. So I think we can call it a day. I think it's okay. Thank you so much, sir, for sparing your time with us and for being here for the talk. We thoroughly enjoyed it. It was like an eye-opener, so much to learn in genetics actually, and we know very little. I think just the surface. As clinicians, we should be like knowing more of it. So thank you so much, sir. And just an announcement for everyone. Next, we'll meet on June 21st, which will be Essentials of Ophthalmic Imaging. That is MRI for an ophthalmologist. I don't think you should be missing that, by Dr. Ravi Verma, sir, from City Neuro Center. So see you all, and a very good night. Thank you so much, sir, again.
Thank you, Shefali. Thank you.
Thank you, sir. Thank you.
Bye. Bye.